great pleasure to introduce Jack Schwartz, who's going to talk to us about a case of signal reconstruction in biology, essentially signal processing. I know Jack Schwartz for 20 years maybe. He was associated with us at IBM. He said this morning, he spoke about somebody whose name is John Cock as his mentor. John Cock certainly was my mentor, and in some sense we have to thank the establishment of speech recognition to John Cock's enthusiasm. He supported it at IBM and got us the money, and we never had to go to DARPA or anything like that in the old days to get any money. We got it directly from IBM because John Cock was in our corner. Anyway, Jack used to work in mathematics and wrote a book on functional analysis. And then he met John Cock, as he told me this morning, and that got him interested in parallel computing and compiler optimization. He even married into the field. And the thing that is remarkable and special for us is that for 2 years, in 87 to 89, he was the head of ISTO, which is the Information Technology division of DARPA, and I came to him at that time and said that the government should establish a kind of a national language treasure, and he had the vision to agree with me and gave money to what is now LBC, and so we have LBC because of Jack Schwartz. Anyway, for the last 10 years he's been also interested in biology. He's a professor at the Courant Institute, and again he's going to talk to us about a case of signal reconstruction in biology.
Thank you. Well, let's see, in inviting me here for this talk, which I'm very pleased to give, Fred told me I could talk about whatever I wanted, but it had to be interesting. So I will talk about whatever I want, and I hope it will be interesting. Let's see, I have a PowerPoint presentation like everybody else. Let's get it started. Okay, so I want to talk about what I think is the great field of science for the coming century, or maybe the coming few centuries, and something that I've always found, since I knew a little bit about it, found profoundly interesting, which is what's going on now in biology, the molecular developments in biology. Since Fred tells me this is not familiar to most of you, I want to just introduce the basic concept so that you can see what the issues are. And then I want to talk about a particular case, a leaf on the vast forest that I've been involved with through a contact at Cold Spring Harbor Laboratory. So let's first think about the basics. Incidentally, am I audible if I'm not?
If you're not, they should move forward.
What's that?
We don't seem to have a microphone. I don't know why we don't. But
yeah. If I'm not audible in this room, either move forward or wave your hands agitatedly, and I will speak louder.
What did you say, Louis? We don't seem to have a microphone. We only have a recording microphone. I don't know why we don't, but.
Okay, but I need a signal if I'm to be reminded to speak louder. Okay, so let me introduce sort of the basic facts, more in abstract terms than other. The biologically significant molecules, the most significant molecules, are polymers. So they belong generically to the family of rubber and plastic. Okay. Those molecules are long chain molecules, and rubber is polymerized by sticking together a lot of smaller molecules. But in rubber and plastic, the molecules that combine are all of the same kind, so they're homopolymers. And a chain like that carries no information other than its length. The thing that is crucially different about the biomolecules, and it's true of all the large biomolecules, is that they're heteropolymers consisting of several different elements. The minimum would be 2, because with 2 you can already form a binary string. And any such string can carry information proportional to its length in the normal sense. So that's the most basic thing that is going on. Now, there are 3 of those large molecules that play critical roles. DNA, which is the basic memory molecule, and think of that as beads on a wire. There are 4 kinds of beads in the DNA molecule. So it's each bead represents 2 bits of information. Okay. Then there's a copied form of that, the RNA, which you can think of as beads, the beads from the wire copied onto a string. They're basically similar, but more flexible, and that plays a role in certain situations. And then there are proteins, and think of those as magnets on a string, of which there are 20 kinds, and those are really copied by a 3-to-1 code from the DNA molecule.
Say it again. Say the last thing.
I say that the proteins are copied from the RNA, which is the RNA is faithfully copied from the DNA. The proteins are copied from the RNA, but the code is degenerate because you use 3 bases, which would be 4 to the 3rd, which is 64. So that can code for 64 things, but it really codes in a degenerate manner for 20. Okay. And, all right. Now, the other basic facts is that if I name the beads +1, -1, +2, -2, which would be a rational way of naming 4 items, the pluses and the minuses attract each other. Okay, so that's a basic physical fact. Okay, and that stabilizes, that attraction stabilizes pairwise combinations, like this molecule, -1, -1, +1, -2, +2, and so forth, attracts more and more strongly over the length the complementary molecule in which all the pluses are changed to minuses, because of the basic fact, the basic chemical fact, that pluses and minuses attract each other. Okay? Normally these are not called +1, -1, and so forth, but they have chemical names, and the first letters you use, so you see the sequence is A, made up of A, P, C, G, which are the 4 beads, as I'm calling. Okay, and this has a crucial consequence. Namely, if you put a molecule of either of these types into a soup of isolated particles, okay, and zipper it up, just zipper up the combination that you can naturally form in view of this interaction, the molecule will copy itself into its dual. Okay, its natural tendency in the presence of that zipper is to copy itself into its dual. I thought of a physical analogy of that. Suppose I took a lot of magnets with little magnets with rectangular holes in the middle, okay, and I lined them up on a flat piece of metal that held them in position, okay. I could put each magnet either plus minus or turn it 180 degrees and put it minus plus, okay. Around the edges of that the long magnetic strip that I formed, there would be a binary pattern in the magnetic field, okay? And then I could hope to stick that into a pile of magnets, and when I pulled it out, the pattern should be reproduced, okay? And that's in terms of a crude analogy, what's going on here. Okay? And that's, so to speak, the basic secret of life. Because that's the great discovery of Watson and Crick, because it gives away, the most basic thing about life is that the processes of life reveal the presence of an underlying pattern. Okay, what you're seeing is clearly not just statistics. Okay, and that pattern has to be copied, or else it will, dissipate. Question?
Dual meaning complementary.
What's that?
Does dual meaning complementary?
Yes. Yeah. That pattern has to be copied. So the most fundamental thing you need is a way of copying that will preserve information. All right. Now, so there are these three molecules. DNA is copied with RNA, and at each copying point there's a kind of zipper molecule that has to act. It actually works as a small molecular-sized little motor that runs along the spring, zipping things up, And so DNA is copied to RNA, and then the RNA actually, the more complicated cells, gets edited along the way, and is then copied into the proteins. Okay, and the zipper molecules, the so-called zipper enzymes, as they're more technically called, are now commercial things that you can buy. They're expensive commercial things, $100 a drop, but they're commercial things that you just buy. And that means that the first step here, the copying of DNA to RNA, you can actually do in a test tube. Okay, you put in a piece of DNA, you put in 4 kinds of beads, you put in the zipper molecule, and it zips itself up. The further steps are more complicated, and it still hasn't been possible to do them in a test tube. So what you do instead is you put the molecules that you're interested in into a bacterium or a yeast cell and make use of the mechanisms already there. Okay. The DNA molecules can be very long. Okay, those are the basic memories. It's I should say GB, that's an error. It's 2.5 gigabases, so it's twice that much in bits. 5 gigabits is the code for the human. For some strange reason, 30 gigabits is the code for the onion. So the onion can boast of being a superhuman creature by a factor of 6, if you want to look at it in those terms. Okay. As the rest I've said. Okay. Now, from these underlying ingredients, what biologists are now trying to do is trace the series of mechanisms which lead from this basic memory capability to the whole structure of the biological world. Okay. The first thing you want to think about is to understand that activity or aim is what living cells must do. Okay, I guess I missed an element here. They have to power themselves. Okay, so there's a whole set of sub-mechanisms that are really just a power cycle in which typically, for example, sugar molecules are broken down and another molecule is pumped up in energy and then serves as the basic internal battery of the cell. Okay? They have to produce their wrappers. Okay? They have to do these various internal copying operations. And then there's a fourth thing, which is that since the DNA molecules are large, they have to be transferred in an orderly fashion without damage to the sequence of bits on the linear molecule when the cell divides. So the cell must be able to rip itself in two, supplying each half with a copy of the basic program on its DNA. Okay? And from that point of view, a living cell can be defined as a bubble containing DNA, RNA, and proteins, able to collect elements from the environment to make more of these things, and able to transfer a copy of its DNA to a new cell copy once it's grown large enough. Okay, so they power themselves, they collect materials, they get to be twice as large, they sense themselves as being twice as large, and then they rip themselves apart, and you have two cells. Okay. They have to have these three basic kinds of machines, the polymerase that copies DNA, the other enzyme that makes DNA into RNA, copies DNA into RNA, and then the so-called ribosomes that make proteins, and then finally they have to have an apparatus of division, which gets more and more complicated the larger and larger the DNA gets. Okay, and in terms of analogy, you could think of it this way. Suppose you had some vital secret on a Peruvian keepy, as it's known, a length of string into which knots have been placed to carry certain information. But this was 1,000 miles long. Okay, and you have to now package the thing up. You have a second copy of it. You have to, next to the first copy, you have to package it up and cleanly separate these two copies. So there's an elaborate folding and packaging and wrapping and twisting and so on and so forth that accomplishes that. And that mechanism is still not, that's the basic mechanism of cell replication. The crucial process of dividing neatly in half, and it's still not fully understood, though a lot is known about it. Okay. The proteins exist within the cell as folded lumps. Here's a typical picture of a protein in its natural configuration. It's actually a linear molecule. It's what you would get if you had a length of string with magnets all along, okay, and you shook it and everything stuck together and you got a folded lump that was still magnetic on the outside. Okay, and that's typically, there are all kinds of different shapes, I think without all that much rhyme or reason, and with different magnetic fields around the outside. Okay. Now, viruses, it's worth distinguishing between sort of various grades of life. There are the viruses. The viruses are sort of minimally, they live modestly, let me say, because they're basically just DNA with a protein coating strong enough to protect it from the environment. Okay? They cannot make proteins, so they can only exist parasitically. They have to inject themselves into a cell that has the protein-making mechanism and exploit its mechanism. More or less, you could say the same thing about a computer virus. It's not a full operating system. So to do its thing, it has to know enough about Windows that it can inject itself into a Windows environment and replicate. It couldn't do that all alone. Okay. But the advantage is they can be very simple because they don't require the full operating system they require. We're going to get it from elsewhere. Now, techniques have been available now for 15, 20 years for reading the sequence of beads from DNA. Okay, you've all seen that on television as the human genome sequence, and you can read the information. And I just give you, this is what it looks like. You probably can't see that too well, but it's just a string of A, C, T, G in some random order. And I show you the start of the smallpox genome, okay, and a similar section determining male development in humans, a similarly long section. The smallpox genome is only 2,000 bases. I forget the exact number, so it's a very small virus. The human is 2.5 gigs of bases. So this is a small excerpt on the bottom of the logic. And the thing to say about these things is that they're really hex code dumps, and they don't have any more, there's no more information directly visible in them than there would be in a hex code dump. So it's not the obvious place to begin without translation software, any more than we could start studying, the Windows operating system by giving you a hex code dump of a million bytes from somewhere in the middle of the Windows operating system. It just wouldn't tell you much. And likewise, this doesn't. You have to know more about what you're looking for. Now, in order to do that intelligently, one needs to sort of think more about the situation at a higher level. So I want to talk now, move up one level from the raw molecules to what's really going on in the configuration model. Okay, cells detect changes in their internal and external environments by changing the molecular concentrations of various things, including proteins, and that has the consequence of a piece of sugar may glom onto a protein and cause it to bend, changing its shape slightly, because everything is magnetic and wiggly. Okay? Now, the way the programming in the system goes is what I now want to explain. The rate of production of every internal protein, and hence its concentration, is controlled by the rate at which the precursor RNAs are being copied from the DNA sources. Okay, so the DNA, say in a human, has 37,000 genes, 37,000 significant regions, more or less, and each of these is being copied all the time at a certain rate, and the rate of copying of these sections is being upregulated and downregulated by the mechanism that I want to now explain. Okay? I have a physical demonstration of what is going on, I think the basic principle is very simple. If I have information along a string like this. Okay, so think of this not as being a uniform white string, but a pattern heteropolymer string, of which a certain section is going to be copied by a linear molecule that will start here and come clanking along and copy off the information, like a reading pad. Okay? And if I now have something else that will clamp to it, like so. Okay? The presence of this clamp can either interfere with the attachment of the copying head at its very beginning and so prevent copying, or because everything is magnetic, it can attract the copying machine, help it attach to the spring, and so enhance copying. Okay, and you certainly have both effects in the biological system, and that way, the presence of certain elements leads to the increase or decrease of other elements. Okay, so you have the basic. You start to sense the basics of a programming situation. Okay, at this level. Okay, now this started to be investigated in the 70s with certain viruses and bacteria because they're small and simple and easier to understand. And a famous case is the so-called lambda virus, which attacks the E. coli germ. The E. coli germs are they're pets, everybody's pet, because they live in our intestines. Okay.
Because they what?
They live in our intestines.
Oh,
that's their natural habitat.
Right.
So they eat what we eat, like a good pet should. And but they're subject to diseases, namely this lambda virus can attack them because the virus is much smaller than the germ. So now the virus, it was discovered that the virus, when it attacks, can do one of two things. Okay, most of the time it just gets into the cell, exploits the internal mechanisms of the cell to make many copies of itself, poisons the cell so as to break it up and get out, and then all these copies repeat. Okay, so it will grow exponentially. But another drastically different thing can happen. Namely, the virus sometimes gets in, doesn't repeat, doesn't replicate, and instead it inserts itself into the bacterial DNA. Because it's just a piece of DNA and it makes a cut in the germ's DNA, inserts itself, okay, and then it's simply there as, as a legitimate part or illegitimate part of the genomic DNA. Okay, and when the germ divides, it will divide, so it's really totally parasitic on the bacteria. And then, 100 generations later, it can release itself and re-emerge. Okay, so all along you have this totally hidden infection with no symptoms whatsoever. And but then sometimes it just, like Dracula from the grave, it emerges. This, incidentally, is a common thing, and it means that all the DNA in the whole realm of life is continually being stirred up by these traveling viruses that go from one place to another, insert themselves in DNA, and then cannot be seen for anything foreign. They look the same as everything else, and the heck's done. And okay, now the mechanism by which this phage virus regulates itself between these two utterly different types of behavior has been worked out in detail. I won't give you the details, but it turns out that the basic mechanism is that there's a little stretch of 3 attractive points along its DNA, okay? And if 2 of them are covered, it will block the one activity, and if the other 2 are covered, it will block the other activity. Okay, so that's a binary switch depending on where in 3 successive positions, separated by a few angstroms, you have placed clamps. Okay, if you have placed clamps here and here, then one behavior will be triggered if you place clamps here and here, and another totally different behavior will be triggered because a different set of proteins will emerge. Okay. Now, what biologists are presently trying to unravel, and it's an immense work. Maybe I'll have a chance to just say how immense in a while, is the details of this mechanism. Okay. One of the most outstanding pieces of work that I know of is relatively recent work by a group at Caltech, who, in a very painstaking way, dissected the structure of one gene in a sea urchin. Okay, and the situation they found is the following. I'm going to illustrate it with my rope. Okay. Think of the gene as this section of rope with the information on it, okay? And the copying machine always travels in one direction along the rope, let's say, top to bottom. Okay. In front of the place where the gene is, there's a stretch that they have analyzed in great detail by cutting it up and otherwise manipulating it, removing sections and very progressively through thousands of experiments. And they found that there are about 40 places along the preceding stretch, 40 places where other molecules clamp on. Okay, and depending on the pattern in which they clamp, which in turn reflects their concentration, that will either, at the point where copying starts, which is down here, enhance the rate of copying or diminish the rate of copying. Okay, and they've worked that out for this particular single case out of thousands in considerable detail. Okay. Various physical mechanisms are involved, and I just want to illustrate one of them, which is particularly easy to understand, to see what's going on. Namely, there's a type of molecule called a leucine zipper, which has a kind of long stem that you see here. When it folds up into a long stem, and sort of think of it as a long stem and half of a pliers. Okay. If you have two of those together, they will make a clamp. Okay. If the clamp is properly shaped and of the right magnetic quality, it can, as you see here, clamp itself onto the DNA. Okay, so there you have exactly the kind of thing I've been illustrating with my rope and clamp. Namely, you have a molecule that can form itself out of two halves. Okay, and when it forms itself, clamp onto DNA at a particular section and either enhance or repress the production of something else. Okay? Now what you have here is a basic programming mechanism. You know, this is a mechanism that is just as fundamental as the flip-flop and NAND gates that you need in computer programming. And it represents, if you threshold it to say, let's not worry about rates, let's say 0 or 1, okay, and think of it as Boolean logic, depending on how whether this is an attracting clamp or an interfering clamp, you would have one of two forms of logic. The output protein of the stretch being copied will be present if both the halves are present, or you might have the negative. Okay? So you have here the basic possibility of programming. You know, this enables programming in the same way that speaking abstractly, that the ability to put NAND gates on a silicon chip enables programming. and it's at about the same level. Okay? And it lets the set of genes present on the DNA function as a program whose execution state is defined by the presence or absence, or more properly, the concentration of the corresponding proteins. Okay? And the programs like this, they have the additive flavor of Markov rule systems in the sense that you can always pitch in, you know, they're very modular, they don't depend on any ordering of statements, and you can always pitch in a few more rules and something will happen. Or you can take out a few rules and something will happen. Okay. And of course, what happens might be very surprising because you're interfering with a circular logical flow. Okay, so in those terms, let me offer a wild speculation, which are always the most interesting things. Namely, how might a cell function as a universal molecular computer? You know, universal in the sense that I want to explain. Okay? So what you can do is use 12 proteins, which would be either present or absent, and that defines a 12-bit state, so it's 4,096 possible states. Okay. You can sense the internal and external environment by activating or inactivating, and from these basic activations, and where activation might be a sugar molecule sticks onto one of these things, changes the way it clamps, and thereby informs the whole system that sugar is present, or a particular sugar is present. Okay? And you would do that for as many as you're concerned with. You know, those are the things that you're able to sense, the cell is able to sense in its environment. It might be things external, like the presence of an external sugar that has been used for power, or it might be something internal that regulates, for example, an internal cycle. Okay? And then if you set up the right, I won't bother you with the details, sequence of logical controls over these 12 bits, okay, you are able to sequence the system. You're able to sequence the system in a completely universal manner through all of these 496 states. So at any point you can take one of two successive states. Okay, and in that sense, for a system of this size, you have something that is entirely universal. So its behavior, it may turn on and off in some totally amazing fashion. Okay. Now, a few very simple things of this kind have actually been built. Okay. It's relatively recent work, but they've been built as DNA molecules that have been expected. One is a flip-flop that was built by a group in Boston, where it can be in one of two states, like the lambda phage, and when it's in one of the two states, each state, as is appropriate for a flip-flop, represses the other and enhances itself. So if you put it in state A, it will keep itself in state A. Put it in state B, it will keep itself in state B. And to switch it, you have to change something in the external condition that will override the tendency not to change, force it to change, and once it is changed, it will remain in the stable state. Okay? So that's a basic element. And another thing that has been built is a three-phase clock, where there are three proteins involved. Each represses itself and enhances the next one in sequence, and this actually cycles through a three-phase cycle of protein concentrations. I think with something like a 10-hour period. So, you know, this is a speculation, but things, you know, a few odds and ends in reasonable accord with it are, in reasonable accord with it have actually been produced. Okay. Now, again, the machinery needed for all of this is you need basically three kinds of copying machines, molecular copying machines: the ribosomes, the protein synthesis. Now, the general structure of ribosomes is known. In fact, that's work it was first published about 2 years ago in detail. You know, a lot was known before, but the full glorious crystallographic arrangement of all the atoms was published for the first time 2 years ago. That's a big clanking thing in molecular terms, consisting of 53 proteins and three pieces of RNA stuck into it. It's one of the most, if we ever lost the ribosome, we would have to start back 4 billion years ago with the whole development of life. It's one of the most essential pieces of complicated stuff to come out of the last 4 billion years. And it probably arose about 3 billion years ago and hasn't changed much since. That's a reasonable guess. And you need the auxiliary zipper molecules for DNA and RNA. You need detector molecules for detecting internal, external conditions. You need structural proteins to stiffen up the cell, basically so that it can tear itself apart in an orderly manner when it's time to divide. You need very small molecules for cell wall maintenance and coating, and you need the basic control proteins that I've been speaking about. Okay, so if you say, all right, I'm going to really put this together, and you know how small a program will do it, or put together this whole operating system, it adds up. You know, it's hard to do anything non-parasitic with less than a couple of thousand molecules. Okay. The E. coli genome, so let's compare that with the facts that are now known. The E. coli genome is about 4.5 million bases and has 4,300 known genes. Okay, so you know, it means the 43 million long bead-long string is divided up into about at intervals of about 1,000 into significant substretches. The human has 2.5 gigabases with about 37,000 known and suspected genes. E. coli detects at least 30 conditions. You can see it switching its behavior. For example, it will respond to different concentrations of various sugars. It will respond to heat and cooling. It will respond to osmotic shock. It's detecting at least 30 conditions as far as is known. So it's already a reasonably intelligent little critter that knows a bit about its environment. The 4,000-state cellular computer that I mentioned on the last slide is probably complicated enough to sustain even multicellular life, which raises another order of complexity, because for multicellular life you have to start with one cell, and basically they have to grow, stay stuck together rather than going into independent business, and specialize. Okay, so you need a program that will do those things. But I think, in fact, that could be, all of that could easily be programmed in a 4,000-state system. Okay. And finally, one of the crucial differences from standard computer technology is that the cellular systems are inherently parallel at all levels from their marker programming on up. you have this long molecule divided, let's say, into 4,000 sections. There's something going on simultaneously on all 4,000 sections. And then you may have, a tissue like a human being like myself consists of 10 to the 13th cells specialized in various ways for their kidney function, liver function, eyes, ears, nose, or throat, or whatever is required. Okay. Now I start to get to the next one. When you have programs, you have bugs. The two things go together. And a bug in the program of a germ cell. Now, where could bugs appear? Okay, well, a bug might appear in cell number 1. Okay, so you start to develop, starting from cell number 1, but there's a bug in the program. And that happens all the time. It just it leads to an invisible failure of development, which is seen as a non-pregnancy or an undetected pregnancy. Okay? And if it happens over and over again, it's infertility. Okay. If you make it to birth, but have some significant bug, it is then manifest as a genetic disease. Okay, and since there are 37,000 genes, there should be at least 37,000 genetic diseases. And in fact, there should be maybe 10 times as many, because each gene might have various defects. So there should be 300, 300 to 400,000 genetic diseases, and in fact, thousands are known. If you have mutations of two sorts that can create bugs. One is just a one-bit or one-base copy failure, where A gets wrongly copied to T, and then that gets propagated in the germline, and you have a whole group of people who are all carriers of a particular disease. But you also have these retroviruses, that are thousands of bases long, that go around here and there, and they go around here and there. They install themselves in DNA, in various patches of DNA, sometimes at random, sometimes with certain preferences, and if they land in the wrong place, they are disrupting something. A rule, they might wreck a rule in a markup rule system. And so that's going on all the time. If it happens to a single cell other than a germ cell, if it happens in a germ cell, you have a genetic disease situation. If it happens in another single cell, it's not important unless it causes uncontrolled growth of that cell. Okay? Because, which can happen in one of a number of ways. Okay? You might have injury to something in the whole sequence of mechanisms that attend to DNA copying, repair, and segregation. Okay, and that's bad news when it happens, because it starts a process. It's like the piece of metal laid on the train tracks that starts a train wreck, because the genomes start mutating and breaking up into pieces and fluttering around, and they should be copied in an orderly manner, and shortly thereafter you have a genetic train wreck. Okay, and you can see it under the microscope that this cell has the wrong number of chromosomes, or one of the chromosomes has lost a big section, big enough to be seen under the microscope. Okay, so that's one thing that can happen. You might have injury to a growth control factor producing uncontrolled growth. Okay, now, about growth, I can say just a basic fact. Starting from a single cell to get to infancy, to get to the stage at which I could survive by being born or go on to be born, I had to divide 39 times. That initial cell had to divide 39
say it again, you have to provide what?
39 successive divisions.
Uh-huh.
And I had to get to size 2 to the 39 cells. Okay? And then for the rest of my life, I had to go on from 2 to the 39th to 2 to the 43rd.
I see.
Okay? And at 2 to the 43rd, things are supposed to stop because I'm not supposed to keep doubling in size every couple of weeks. So the mechanisms of growth are supposed to turn off after, somewhere in the program it has to say due from 1 to 43.
Due from?
Due from 1 to 43 and then stop, And, but I have to retain in certain cells the ability to grow because I might be cut. So I have to have the cells have the ability to sense an injury and repair themselves by regrowing. Okay, so there has to be a control mechanism of growth. But if that becomes uncontrolled, it's very dangerous. Okay. So you can see that certain bugs in a single cell, can develop into large clones of the barren cells that have may have devastating consequences, especially if they lose their tendency to clump together. That's a matter of secreting the right glues on the outside that normally keep cells of the same type together in most cases. If they lose that, they can start wandering all over and get into your kidneys and start destroying your kidney function. So that's what's happening. Now, so that's really the known underlying mechanism of cancer. And it's known that the genomes of tumor cells are often damaged quite badly. Not necessarily, it might be something, just one base in the growth control molecules, but often it's wholesale wreckage. There's a lot of interest in the close inspection of tumor cell genomes to see what has happened. Okay, the closer the better. And in fact, it would be even better to do this earlier. For example, if your family had some known disposition to cancer, you might want to, before marrying somebody else, you might want to undergo a genomic inspection so they could see this combination is dangerous. Okay. Now, there are tools that are available. They have become available for such inspection. And I want to talk about one of them, which is the DNA microarray.
DNA what?
Microarray.
Microarray.
Array. The DNA microarray, which in use looks like this, and it's a wonderful combination of laser technology and ideas from computer technology with the biotechnology. Okay. The way it works is that on such a chip, you deposit in a controlled manner, let's say on 100,000 spots. Okay, on 100,000 spots on the chip, you deposit 70 base pair long pieces of DNA. Okay, every one of them different in a known fashion. Okay, so it's like a memory. You know, it's exactly like a ROM, except the things that are being remembered are these stretches of 70 bases, you know, 140 bits. Okay, and you are able to send the manufacturer of these things a file saying, I want this pattern, you know, 100,000 times 100,000 times 140. Okay, so you send him a 10.4 megabit file, and he sends you back the chip with the DNA on it. Okay, the way it's done, you know, it's an astonishing piece of technology. It came in 10 years ago or so. And it's done various ways by various companies. This company, Nimblegen, does it using the Texas Instruments optical controller chip and actually writing the chips independently. So they'll put, you know, the successive bases on the chip and energize the spots with this Texas Instrument optical chip, add at a given place, add one more base, and in that way build up the completely controlled sequence of 70 base parallel strings to order. $750 a piece. It's an amazing price. But I'm not sure that may be a discount price. That's what they're selling it for to people at Colson and Harvard. Okay? Now, to use a chip like that, what you do is take something that you're interested in. Okay, you're interested in something, and you say, it may contain a section dual to, and hence attracted to, one of these 100,000 things. Okay, so you take the thing you're interested in, you attach a little red phosphorescence molecule to the end, paint it on, it will stick in the one place out of 100,000 where there is something matching, and then you can see it. You put it under a phosphorescent wire and see it. Okay, and that's become a standard technique, which is sort of, it's in its normal application, it's parallel at the level of 100,000 pieces of sensed information with each experiment. And it has started to produce an immense flood of information as you know, readings of 100,000 values come out from each of these experiments. Okay, maybe I'll skip this. It's all interesting. Okay, let me now talk about the particular project I'm involved with and some of the signal processing issues involved. Okay, I'm working with a group which I happen to meet and enjoy. They're a Cold Spring Harbor lab, and their specialty is human cancer, because of breast cancer. And they're trying to find out more about what's going on. Okay, it's a research rather than therapy, medical group. Okay. And they've put up this procedure, which I'll describe to you. You get a reliable 3% DNA subsample, just to reduce the sheer physical weight of DNA that you have to deal with, because the whole human genome is a little too large. You cut it in a known manner. I won't bother saying exactly how it was done. And you pump up the things, and you then pick out the shorter ones. Okay, and you have about 135,000 shorter pieces to pick from, and you select of those by computer 85,000 which have highly distinctive 70 base pair long sequences, because you don't want things that will accidentally stick to the same spot. Okay, so in the detailed design of this experiment, they're trying to get as clean a signal as they can. Okay, you tag them in the normal way with red or green phosphorescent markers at the end. You get this company to prepare the chip, and then you do a comparative hybridization, normally of two things, so it's done comparatively. You have a red tagged substance you coat on, and a blue tagged substance you coat on. You paint them both on, and then you compare the intensity of red and blue at each single spot in order to get a normalized reading. Okay, now this, I hope you can see it, this is what the data that emerges from such an experiment looks like, okay? So this, what you're seeing here, left to right, is just the index of one of 85,000 probes. And the section you're seeing is about 900 of 85,000 probes. So this, among other things, should give you a sense of the size of the data processing problem that's involved. Okay? And the signal is the log of the ratio of red to green as sensed in each spot. Okay? And what you're comparing is, for example, a normal cell, DNA from a normal cell from a person to DNA from a cancer cell in the person. Okay? In fact, I think what you'd really rather do is, rather than waiting for this train wreck to occur, is you want to, what you want to do, say, is compare a normal cell in me to a cell taken from a mole on me that my dermatologist might say, that looks a little funny, I want to keep an eye on that. Okay, and, but I won't cut it off right now. Okay, and if he had a better tool for looking at it, he could look into the DNA and say, oh, you know, there's a section very near, or perhaps in the middle of the growth gene that has been deleted in the cells in this mole. Let's not wait let's cut it off right away. Okay? And so that's the sort of thing one is looking for. Now, what you see here are, let me try to explain the data. I'm sorry it's not better visible here. It might make sense to turn on the lights for a few minutes.
Well, maybe.
Yeah.
It's fine.
Over there, probably.
Let's see, this will possibly cause the
Well,
room to
something is
explode.
going to happen if you touch it. I guess nothing happened. So I don't know.
No effect.
I don't know where we have the lights. Maybe somebody.
Yeah, there must be some way of turning off the lights.
But I mean, we see it all right, don't we?
Yeah, okay. Can you see it all right? Yeah, okay. You notice, for example, in this particular stretch of genome, so one up there, there's something strange about it. As a matter of fact, if you look at it, it's the same little stretch in enhanced things. You see that there's something strange about two successive probes in a row. Two successive probes in a row along the genome, which are separated by about 30,000 base pairs, both have aberrant readings. Okay, and that points the finger of suspicion at that 30,000 base pair long section, which with probability 1/2 contains a gene. Okay, so at that point you can go to the tables, you know, the maps of known gene locations and say what's there. Okay, and maybe come to some conclusion. Okay, if you look at a case like this, and these are all normal to tumor comparisons you're looking at, you see here that there really has been a train wreck. Okay, because the signal is coming along like this, and this whole section up to here has somehow changed. Okay, and that's a section that probably contains 100 genes. In fact, it's more than that. This is that's about 450 bits. That probably contains about 200 genes. That section. So something has happened. You know, these cells have either lost 200 genes or 200 genes have been doubled. So, you know, this Markov program 200 of the levels that are controlling it are wrong. And this is already at the train wreck stage. You know, so, and those things are easier to see. So that's what one is looking for. Now, they're trying to promote this to for example, a tool that could be used medically. And, you know, somebody who's doing this en masse doesn't want to have to sit there and scan through 85,000 data points, even graphically. So you want to reduce this to a much more compact signal. What am I doing for time?
Well, we're not doing too well, but we have another maybe 10 minutes.
I'll try to wind up. So you're trying to read this, you know, the signal out of this obvious field of noise. This is something I'd like to discuss with you if there were time. What conceptual rules you want to use? Now, you're always, it seems to me, trying to use the assumed characteristics of the signal and the noise in every such situation, speaking abstractly. You assume that they have certain characteristics and you're trying to make them an optimal separation of signal from noise. And it is optimal when there's no signal in what you have called the noise. Okay, so you're trying to recognize, you're trying to get an algorithm that will produce something called signal, something called noise, and the noise should be pure noise. Otherwise, there's no signal. Here, because it's assumed that what's going on is that two things. Big sections of the gene are duplicated, falsely duplicated, or deleted. So there are big lesions and there are small lesions. Okay, so you assume that the signal, it's reasonable to assume that the signal is a step function punctuated by occasional little peaks. Okay, so that's the assumption about the signal. And then that suggests, you know, a heuristic way. One can either get fancy about these things and say, you know, I'm going to use some mathematically ornate method, or I prefer, in fact, to be crude in these situations. And so I use a crude algorithm, crude heuristic, and what that algorithm winds up saying, well, it's what you see as the line drawn through the middle, that is the signal, and everything else around it. The middle thing shows the difference is the noise, and that tracks reasonably well over rapid changes in the pattern, because it's a moving average type of method, but it's tuned, and we'll take the time to say how, so that it does better than moving average would on tracking a signal like that. You know, tracking a presumable signal like that. But the output I just showed you is not good enough for the busy researcher who still doesn't want to look at that smooth line. They want to have a list of features. You know, so you have to flatten drastically and say, I'm going to reduce everything to horizontal lines with jumps at certain points, and I'll give you the list of jumps, and you have to do the rest. Okay? In fact, in these cases, that produces lists of maybe 150 jumps. It does, so you see here that more drastic flattening of the signal, and it does, I would say, about as well as the first cut. If you look at the residual noise, it doesn't look significantly less like noise than it did previously. Okay, so this entitled me to settle back in a fog of self-congratulation until a colleague came up with another method and said mine is better. And I didn't want to say, no, mine is better, because I like the looks of it better. So there really then gets to be a question of objective performance assessment. So given these signal reconstruction algorithms in general, how can you establish a rational way of saying method A is better than method B? Where by rational I mean more than merely aesthetic. It's because I could stand there with my colleague and he would say, my method is much more sophisticated than yours, and I would say, exactly, my method is much more robust than yours. So here's a cut at it. One thing one can do is take this, I'll put it as simply as I can so it doesn't take a lot of time. You take the signal noise separation that you've read out of the data, and then you replace the noise with artificially generated noise, and that gives you another quasi-original signal, and you read that, okay, and then you can assess the stability of your reading, okay, and I'm proposing the criterion that readings which are more stable have some claim to be considered preferable, because at least they have the virtue of consistency. Okay, I ran a couple of runs like that on the method I've shown you, and I'll show you some of the numbers. It turns out the numbers suggest that this technique is, for example, 80% reliable in reading wide features and something like 60% reliable in reading narrow features. So that's about where things stand, and I'll just stop. Take any questions.
Anyone? Any questions?
Just then a final word to the undergraduates and graduate students present. Don't work in any other field, work in this field. because there's going to be a century of wonderful discovery, the skills that you have in signal processing and grammars and everything else are all relevant. The biologists don't have enough support from computer scientists. For example, Cold Spring Harbor, which is where a Nobel Prize is regarded as an ordinary event, has a very half-baked computer activity. They just don't have the people. So, and I'm sure that that's the situation in many places, so there really is more synergy.
Is the point of view that you're espousing here or describing sort of universally accepted? I mean, this is a point of view of a computer scientist looking at biological phenomena and have hoped to...
More and more it's received doctrine, though it doesn't you know, the fact that things might happen in a certain way, that you can demonstrate this in a simulation, that something that adheres to reasonable restrictions is capable of certain behavior, doesn't tell you all that much about what is really going on. You know, even any more than a couple of papers by Dijkstra on proper programming tells you what's going on in the Windows operating system. You know, this is stuff that has accumulated over billions of years, and it's so. In fact, what that says is it may be much easier to construct biological systems that mimic, you know, patterns of growth and so on and so forth, than it is to find out how they really work. Because to construct them, you just have to understand the basic mechanisms, and to understand what's going on, you have to understand the actual mechanisms. From the ultimate medical point of view, you know, if you go to your doctor and he says, you know, doctor, I have a disease, he says, I don't understand what's going on, but I can replace you with a very acceptable artificial system. That's not fully satisfactory.
But the idea that life is a program
yes, no,
is something that's accepted.
I would say
and doctors will be computer
more
scientists.
and more accepted in more and more detail over the last decade. And I don't think you'd find a lot of argument. Let me answer that a little crooked. I think the issue for the computer scientists will be what are the key biological problems, because the computer science will be a handmaiden to the biology. So it's whatever the customer wants. Okay. The really interesting things are that will be biologically will be the you know, if you think of the protein mechanisms that I described as being some kind of complicated Markov, you know, think of it in the human being, it's a Markov system with 37,000 rules and genes, each rule having 40 inputs from somewhere else in the system, and those 40 inputs being combined by some unknown Boolean formula. Okay, well, that's what you have to find out. What are the details there? Okay, so the biologist has to think of the experiments that will probe into that and that are feasible. Okay, so I think what will happen in biology is it will turn more and more into big automated science, in which you put the experiments will have to be done on some sort of industrial basis. You know, you put a substance to be tested or a cell to be tested in one end of a giant machine and get the data out the other end. That's what happened in the Human Genome Project. You know, they had floors full of sequencers, and then some enormous flood of murky and muddy information is going to emerge out of the readings taken, and the computer scientists will have to help make sense in organizing and making some sense of it.
Is that the chip by any chance, the micromirror?
Yes.
The one that's used for projection?
Yes.
The one that almost died because it didn't produce the print?
Yes. Yeah. So it's
I came so close to dying.
Yeah. It's served a useful role. Okay. Thank you.
Thank you.